Okay, class. So this is lecture 5 PowerPoints, which is the first part of the mutation section that we're going to cover. And then there's a mutation 2, which is what we're going to call lecture 6. So this is lesson 7, right, which is the beginning of the lecture 5 mutation PowerPoint. So hopefully that's straight. Um, and I think I need a new naming system. Anyway, so you probably know this. A mutation is a change in a gene, right? The genetic material, which is in most cases DNA. I mean, if we're looking at a virus, it could be in the RNA if it's an RNA virus, but generally we're talking about DNA. And then you can have two different kinds of mutations. So you can have a mutation in your germ line in the cells that produce gametes, and that's so if it's in cells that produce gametes, then it's transmitted to your offspring through the production of gametes. And that is the source of variation. And then, of course, evolution means change in the allele frequency over time. So these things is what produces the variation and the difference in allele frequencies that we see in populations. Okay, so that's a germinal mutation in your germline, in your sex cells, your gametes. Okay, and then we have somatic mutations. Those occur in your somatic cells. So just hopefully this is a refresher, but your somatic cells are all of your body cells, like soma means body. So all these are all the cells in your body except cells that produce gametes. So this would be like heart cells and lung cells and everything except sperm and egg in humans and most animals. All right, so this is not transmitted to descendants of like your offspring. It's only transmitted to descendants of cells. So, like, let's say you had a mutation in your lung cell. Uh, that mutation could give you lung cancer, and it would be passed. So, let's say that the letter A is converted into a G and in some gene. So, A is now a G, that would be passed on. So that mutation would be passed on, A to a G, to all of the descendant cells from that original somatic cell. But you wouldn't be able to pass this on to your offspring unless it occurred in the cells that made sperm and eggs, the gametes. All right. Point mutations are mutations. So get you some note cards or whatever, make you a crossword. Point mutations are mutations that occur in a single point in the DNA. So a single base pair. Um, I mean, it's generally not a small group because we could refer that to as insertion or deletion. But I guess you could have multiple point mutations. So let's say if you had a and a C and a G, you could have this A converted into a C and this C converted into an A. And that would be multiple point mutations, right? In a small group. So they would, no matter what, you would have to call, they would only occur between adjacent pairs. So this, you could call this a point mutation, but if it occurred over here as well, this would be a separate point mutation. So we can substitute bases, right? We could put in, instead of an A, we put in a G. Instead of a T, we put in a C, or whatever. We can insert things. So, like, let's say we have AGC, and you get a mutation that makes it AAGC. This would be an insertion. You could have a deletion where we remove one of these. So that would end up being AC. This would change the reading frame. This would also change the reading frame. And we talked about the reading frame in the previous chapters when we were talking about replication, transcription, and translation. So insertions means we're adding nucleotides, deletions means we're deleting nucleotides. Pretty self-explanatory. This is just an example of a base substitution. I kind of drew it up there, but like so in this case, a GC base pair would be mutated to an AT. This would be a point mutation because it only occurs in a single point. Insertion, like shown here, you could have this AT base pair inserted in the middle of the sequence. And so, again, that would affect the reading frame. Deletion is removing nucleotides. So in this case, we would take this. GC, which one is it? GC, this one. I guess it could be either one. Those would be removed, and then we would have a new sequence here. Again, this would cause a frame shift mutation, which is really bad. Oh, and I should mention that insertions and deletions are called indels. It stands for insertion and deletion. Makes sense. All right, so this is a review. I mean, should be a review from previous biology classes, but it's also a review from what we learned in previous chapters. So remember, the purines are A's and G's. Those have two ring structures that we talked about. And I had you guys draw those in your study guide, your problem sets. And then we have the pyrimidines, which are T's and C's, and those are single rings. Remember, this is the number 1 carbon, number 2, 3, 4, and 5. This one is 5. All right, so when geneticists talk about mutations, we refer to them as transitions or transversions. So a transition is replacing one base pair with another base pair of the same chemical category. So if you replace a purine with a purine or a pyrimidine with a pyrimidine. So, for example, remember A's and G's are the same. So we're replacing this G with this A, and those are both purines. So they're the same chemical category. And then we're replacing this C with this T, and those are pyrimidines, and those are in the same chemical category. So this would be considered a transition mutation. Transversions are replaced with a different chemical category. So in this case, we have G here. That mutation is replaced with a T. So G's are purines, T's are pyrimidines. Those are different chemical categories. And then the C and A are different. This is a pyrimidine. This is a purine. All right. So there's 12 different kinds of base substitutions that occur in DNA. And then this is just a convenient graph to show you. So transversions are shown in blue. Transitions are shown here in green. So this one, and so on. So we have 4, 5, 6, 7, 8 transitions, and then we have 1, 2, 3, 4 transitions. So statistically, transversions occur more frequently than transitions. And then these are purines, and these are pyrimidines. Double ring, single ring. All right. So there are different kinds of mutations that can occur in, remember, polypeptide means many peptides, and a peptide is essentially another word for an amino acid. So this is basically saying a bunch of amino acids linked together, and of course that is a protein. So mutations that occur in DNA that can affect the protein structure that we talked about in the previous lecture, we can have synonymous, missense or non-synonymous, conservative and non-conservative, and then nonsense mutations. So let's go over each of these. So synonymous mutations can change a codon into another codon for the exact same amino acid. Remember we talked about the genetic code as being redundant because there's 20 amino acids, but there's 64 possible combinations. And so that means that more than one codons, there's 64 possible codons. That means that there are more codons than there are amino acids. So that means more than one codon has to code for the same amino acid. The example here is that we have what we call the wild type gene, or the most common gene. And in this instance, this is codon 1, codon 2, codon 3, codon 4. And then this is the protein, that the amino acid that it codes for, and then these would be linked together with peptide bonds. So here we have a mutation. That mutation has converted by substitution. And remember, synonymous means it's the same amino acid, right? Not the same nucleotide, because that wouldn't be really a mutation. So in this case, this T was converted into a C, right? So this three-letter code, which this is showing in DNA, right? So this would be in RNA. So in this three-letter code codes for arginine. This three-letter code also codes for arginine. Because they're the same protein, right? The same amino acid, the protein structure doesn't change. And so this is a mutation that's not really going to affect anything like color or height or anything like that. But it still is considered a mutation on the DNA level, even though it doesn't change anything. And that's why it's called synonymous. Missense mutations they change one codon into another codon for a different amino acid. So we have our example again here, wild type. Here we have this C that codes for arginine, and then it's mutated to an A, so this codon would now code for serine. And because these are different amino acids, it could affect the shape of the protein and cause it to be non-functional or expressed differently. So that's what we call a missense mutation. So make a note card or flashcard and just learn the definitions. This is how geneticists speak. All right, so if we have a missense mutation, so missense mutation is, remember, it's one amino acid is getting converted into a different amino acid due to a point mutation. So we have conservative mutations, conservative substitutions. Remember we talked about amino acids having certain chemical properties. So we could have amino acids that are positively charged or negatively charged. They could be acidic or basic. They could be hydrophobic or hydrophilic. So let's just imagine that we had one amino acid converting into a different amino acid. And again, I don't want you to memorize the amino acids, but if you look in your book, there should be an amino acid table. So I just browsed through the book and I don't see an amino acid table in there. So I just pulled one up online. There is definitely one in the Campbell biology book that I use. And I'm surprised it's not in this genetics text, but no, I don't want a school license. Um, so I'm just showing you like these amino acids are electrically charged on their side chains, which there are groups. And so we have arginine, which is positively charged. Uh, Lysine is also positively charged, as opposed to aspartic acid and glutamic acid, which are negatively charged. And then we have polar versus non-polar, uh, hydrophobic, right? And then special cases of side chains. So anyway, the point that I'm trying to make here is that these um, conservative substitutions replace one amino acid with another chemically similar one. So if you had a positive amino acid in your protein, let's say that, you know, this one's negative, and this one's negative, and this one's positive, and this one's negative, and this one's positive. I'm just making it up. This one got mutated to another positive amino acid. It wouldn't change the structure at all. Um, well, generally not, because it would have the same effect on the folding of the protein. And we kind of went over this previously. So anyway, uh, like glycine for alanine, for example, both are nonpolar, and so there's likely not to be much of a difference between those two. Then we have non-conservative substitutions, which replace an amino acid of one type with an amino acid of a different type. So in this case, you'd have a positive replaced with a negative, a polar replaced with a non-polar, and so on. And I mean, obviously, non-conservative substitutions are more likely to be really bad uh, in affecting the protein structure and function because more likely than not, they're going to change the shape. And the most important thing in biology is shape. And I stress that with my 181 students all the time. So a nonsense mutation is a mutation that turns a regular amino acid into a stop codon. Remember, these are UAA, UAG, UGA. So anyway, this would be UAG. This is the coding strand, which is also the non-template. So this would be the RNA, right? 5 prime, 3 prime. This is, sorry, 3, wait, wait. This is the template strand. So this is 5 prime. This would also be 5 prime. 5 prime, 5 prime, 3 prime. So this would be UAG in the mRNA, and that would code for a stop instead of this, which is AAG um, codon in the mRNA. And then obviously having a stop codon interrupts the remainder of the polypeptide chain being made, which is going to have a detrimental effect on its shape and therefore a detrimental effect on its function. So premature termination has a big effect on protein function. Makes sense. All right. So, like I said, I'm not going to make you guys memorize the genetic code. I'm just putting it up here. I will give you it on the test. You see, if you took the quiz on the protein, you'll see that I included that in the quiz as well. So it's not going to be a surprise. You're gonna get the genetic code. I don't want you to memorize this. You got other things to learn. Okay, reading frame. We already went over this once. We'll do it again. This is the original DNA, right? And this is the probably the coding because it's been divided to 3. That's usually how it is. Which is the coding strand, remember, has the same code. So this would be A, C, G, and then we replace U's with T's, and so on. So we have this reading frame. But if these letters got mutated, so we got a deletion here or whatever, then it would change the reading frame, right? So we kick it over by one letter, and then it's read in threes, right? Because that's how codons are read, and that's going to change all of the amino acids, which will change the shape of the protein. And then this is the third reading frame, so we kick it over two. And so one is the normal reading frame, two is when it kicks over one, three is when it kicks over two. And if we kick over three, like if we went A, C, G, then it would go back to the original reading frame. This would just be deleted out. So this is really bad. This is also really bad. So you don't want insertions or deletions in your DNA. That can really screw up your proteins. All right. So you guys hopefully know this. The start codon is where the ribosome starts reading. Remember, it's amino to carboxyl. So if this is the coding strand, this is A. Remember, we have the mRNA is written the same as the coding strand. So we have AUG, which is the start codon, indeed. So in AUG, remember, this is the coding strand, so I'm just converting it into mRNA. This codes for methionine, always. GCC codes for alanine, ATG codes, so AUG codes for methionine, CTC codes for leucine, and then this is a U. CUC codes for leucine, and then GUG codes for valine. And then there would be a stop codon in here somewhere. It's just not being shown. So once you get an insertion or deletion, and that's what indel means, after the start codon, it changes the reading frame. So mutations in here aren't going to affect anything. It might affect a bacteria if it's the Shine-Delgarno sequence, but usually the mutations have to occur downstream of the start codon. And so that's basically what's showing you here is that this G is taken out, right? And we change the reading frame to this. And that's going to change all the amino acids downstream from there. This amino acid, and that amino acid. All right. So that mutation that changed the reading frame is called a frame shift mutation. Here's an insertion. It's still going to have the same effect as a deletion, right? It's going to change the reading frame. And this causes major impacts on gene functions. I don't know why I have so many slides on frame shift, but hopefully you get it. And then another thing is, I might ask you on the test, like you have a mutation in groups of 3, and we already talked about this. It's not going to change the reading frame. It's just going to go back to the original frame. A lot of mutations don't have any effect on the phenotype. So synonymous mutations, remember those are mutations that are for the same amino acid. Most mutations are in the introns. So remember we have exons that are linked together, and then intron. And these get spliced out. So any mutation in the intron besides that important A, right? And then the intron-exon junctions. Any other mutations besides those two places isn't going to matter because this is going to get thrown out anyway. Remember, it's just allowing the shuffling of exons to make more than one protein product for the same mRNA transcript. So mutations can affect, what else did I miss here? Oh, most mutations are in the intergenic region. So what that means is, like, let's say this is a gene for beta hemoglobin. And then you have another gene here for, I don't know, collagen. So I'm trying to stick to things we talked about. in between there are called intergenic spacers or IGSs. Mutations in areas that aren't actually genes aren't going to, well, I don't want to say this because scientists are now finding out that this, which they thought was junk DNA, actually codes for areas that can control gene expression. So this is a question mark, but just for now, we can say that most mutations in intergenic regions are not going to have effect on the phenotype, which is true. So we can have mutations in promoters too, and that can eliminate or reduce the transcription of a gene. Remember, we have all these transcription factors that have to locate regions and bind to them, like the TATA binding protein. So if this is mutated, this wouldn't recognize it. We wouldn't have the transcription factor present, and that wouldn't be able to recruit the RNA polymerase. And if we can't recruit the RNA polymerase, we get no gene expression. Yeah. So it would stop the initiation complex. So where do mutations occur? Well, we term mutations mutagenesis, right? Genesis is birth or beginnings, and this is for mutation. So what is the beginning or origin of mutations? And the answer is there's lots of them. So you can have what we call spontaneous mutations. These are mutations that occur naturally uh, due to normal processes within the cell. Um, and we'll talk about what can cause this. And then we have mutations that are induced. Those are generally caused by environmental agents like sunlight or chemicals like smoking or whatever, eating barbecued meat that can increase the rate of mutations. So let's look at spontaneous mutations first. So some of the spontaneous mutations are tautomeric shifts. We can have depurination, which is exactly what it sounds like. D is to take away, and a purination is a puring. We can have slippage of the replication machinery that can cause repeats in the DNA. Remember, all these mutations have to occur in the DNA because if they occur in the RNA, no big deal. You just get another RNA transcript. So these are all DNA level mutations. All right. So tautomers. So each base appears in two different forms, and it has to do with the shifting of electrons within it. So we have adenine. This is the most common form that adenine takes on, which is this amino group right here, this NH2. But this hydrogen can migrate here. And when it does, it forms this shape. Right? Cytosine, normally it's like this, but this hydrogen can migrate, right? And so we have this form. And so these have different shapes. Just so you can tell. Same thing with guanine and thymine, right? We have this ketone here, and then that shifts to be a hydroxyl group. So this goes here. And we can have the same shift in this ketone here. And if you don't remember this stuff, it's fine. So this hydrogen goes here and then forms a hydroxyl group. So this shape is different than that shape. And then they go back, right? So that this is just electrons and atoms shifting and making new bonds, right? And they're going back and forth between these tautomeric forms all the time. But that can be a problem because these bases are shaped differently and they're different tautomeric forms. The polymerase might not recognize them for what they are. This is just showing you what I just did. All right. So the common tautomers, like we just showed, I just showed you, and I don't want you to memorize the chemical groups, right? What I want you to come away with is what the mutations are. So here we have cytosine, and we know that that pairs with guanine. So we know that C pairs with G, and it has three normal hydrogen bonds, just like we would expect. And you're going to have to draw this out in your problem set that's due on the pretty soon. And then thymine, of course, pairs with adenine. So we have T pairing with A, and here's the two hydrogen bonds that we talked about. So that's just normal, right? That's normal DNA base pairing. When we have tautomers, and that's shown here in green, right? We talked about the tautomeric shift. So this is the rare form of cytosine, and then that pairs with adenine. So now we have C pairing with A, and that's two bonds, right? Two hydrogen bonds. That's a mispair, right? And it's just caused by that tautomeric shift. We can have the normal tautomer of, so here, normally would have a double bond here. Normal cytosine, and that pairs the rare form imino is what it's called. It comes from the amino group variation. And anyway, so we have C pairing with A here, where this I'm just going to mark this as an asterisk as the rare tautomer, and this is an asterisk as the rare tautomer. So again, these pair together, and again, this is a mispair. So it can go either way. We can have a shift in the C, and that would pair with the normal A, or a shift in the A, and that would pair with the normal C. Same thing with this is the enol, and it has to do with the ketone group. So anyway, this we have the rare form of T that pairs with G, and this makes base pairs, three hydrogen bonds. Sorry, not base pairs. And then this is normal T, and it's going to make three base pairs with the tautomeric form of guanine, G. So we can have T accidentally pairing with G in this tautomeric form, or the tautomeric T pairing with normal G. All right. So these tautomeric shifts are going all the time inside our DNA right now, and these shifts lead to transitions. They cannot cause transversions, and so I'm gonna show you why that is and how. But so we have a normal GC pairing, and that pairing, remember the C can pair with an A and so on, and that's going to lead to this G becoming an A and this C becoming a T. Right, and remember that because it's so A's and G's are purines, so they're the same chemically. And if you haven't got that down by now, you probably should study that. All right, so here's an example. This is the parental DNA double strand. Normal GC base pairing here. We have replication that occurs. This is where the tautomeric shift, so it's an asterisk. Remember, the rare tautomer of G can pair with T. And we have the normal strand here. So the other strand, we don't have a tautomeric shift. Remember, this is rare. So this might occur, and then the polymerase would incorporate a T, thinking that that's what it's supposed to pair with, this tautomer G, which is right here. So here's the tautomer G, and it would put in a normal T. All right, so now we have, so this is the first round of replication. The two daughter strands now have G and T paired. This one has G and C paired. and then we go through another round of replication. So here, when these two strands separate, this G here is going to pair with C, right? And that's the original pairing. And then this T will pair with A, so this is a mutant. And then this is normal, right? Both strands are going to be normal. So this G is going to pair with this G is going to pair with the C, and this C is going to pair with the G. Right. So in the end, we're going to get 3 wild type and 1 mutant out of this tautomeric shift. But you can also notice that our original pairing GC is now becoming AT, and that is a transition. Right. It cannot cause a transversion. Okay, so we can have depurination as well, right? So here's DNA. Remember, this is a nucleotide, so this is a monophosphate. It has a single phosphate group, 5 prime, 4 prime, 3 prime here, 2 prime, 1 prime, which is the base carbon, which is shown here, right? And so in this case, this purine is cleaved off. Remember, purines are double ringed. So this depurination right here, this cleavage removal, and then of course this is a hydroxyl group, leads to mutations during replication because um, if you're missing a base in the strand of DNA, it's not going to know what to pair with, right? It doesn't know what this was because the base is gone. So depurination is common, but it's when this happens, generally it uses the other nucleotide to tell what it is, and then it can replace this. All right. So you can also have replication slippage. Replication slippage can lead to usually it's deletions, but also insertions. So, and this is usually done because of long stretches of repeat. So in this case, this is a repetitive CT, 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 CT stretch of DNA, and this can cause it to buckle out, right? When the template strand loops, then this might not be copied, so it's left in the original strand, but in the other strand, right, it's not mutated. So we have an insertion here. Sorry, this buckles out and then it goes back to normal. So here's where we have the strand. This strand is replicated without the CT pairing, so we're missing the CT pair here, and then again here. So this causes a 2 base pair deletion, and then this is the normal strands because this goes back the way it was. So the the DNA polymerase, and this is DNA polymerase 3. Usually we have these dinucleotide or trinucleotide repeats, um, like AT, 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 and these are usually the sites of mutation, and they usually expand or contract. And scientists quickly realized that this replication slipping was creating more and more repeats at certain regions, or reducing the number of repeats in certain regions. And so scientists use this information uh, to for forensic identification. And So I just went to DNA interactive.org, dnai.org, and then I clicked on applications and then human identification. So this is how we do DNA profiling, and I can just show you. So Alec Jeffreys is the first person that actually, he's not the inventor of DNA fingerprinting, but he's the first person to use this and to publish it and it be used in a criminal trial. So we have variants in our population of these repetitive elements. These markers are used by the FBI. They're called the CODIS markers or CODIS loci. And so these are the names of them. I don't want you to memorize this. I'm just showing you this why this is can be useful. And so these are short tandem repeats, right? And we talked about those as lines and signs. And that in this case, they can have 9 or more different alleles that can be detected. So if we look at this region, this variable region, we have in this case 1, 2, 3, 4, 5, 6, 7, 8 repeats, and in this one we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 repeats. And so this person's genotype would be 911 for that particular region, that particular locus. And I'm not gonna. We'll cover this later on. I'm not gonna get into it, but we can look at the number of repeats by doing a genotyping. So this is old school genotyping machine, and essentially it's going to read what the nucleotides are, and it'll put peaks there where those primers are located. So in this case, we could say, okay, this person here is a 1214. That's because they have 12 repeats in one chromosome and 14 in the other. This person's 2830 at this loci because they have 28 repeats and 30. They're homozygous at this loci. They both have 9 in both of their chromosomes. And then again here at 10. So if you have 13 of these, you get a pretty good picture of whose DNA that belongs to, who was at the crime scene, who the baby's daddy is, that sort of stuff. All right. So anyway, that's how we can use replication slippage. Now, this also can cause problems in DNA. And we're going to talk about this later on when we talk at chromosomal mutations. But there's this replication slippage can create these long strands of repeats, which can make the DNA fragile. So it's called Fragile X syndrome. And that's due to repetitive elements in DNA. And then we talked about this. So I showed you in the drawing. It's how it loops out, and usually we get a deletion, which I showed you in the previous. Uh, the newly synthesized strands can also loop out, and then that creates an insertion. I don't really care that you know that. I just want you to know that the repetitive elements cause DNA replication to slip. The polymerase slips, and that can create either insertions or deletions. And remember, insertions and deletions are called indels, and indels cause frame mutations, which are really bad. You don't want those. Okay, here's Fragile X. So this is a form of inherited mental impairment. It's detected by, you can visually look at it through staining. It's on the X chromosome, and it's due to the expansion of a triplet repeat, right? Like AGT or whatever, over and over again. So the gene itself in Fragile X is called FMR1, And so normal has about 30 repeats. The sequence is CGG. Um, it's in the first exon, right? So this is the actual coding region of the gene, and that's why it has an effect here. So normal phenotype can have up to 200 repeats. Um, it slips when it's dealing with the repetitive elements of greater than 50. And so the individuals with Fragile X syndrome, when their DNA is sequenced and looked at, generally they have these repeats that exceed 1,000 repeats, and that causes them to develop Fragile X syndrome. And there's really not much you can do about it because it's just repeating the same three letters over and over, which is increasing the size of the protein, which obviously affects its shape and causes downstream issues. Okay, so you can also get mutations from the environment. Um, and so these were what we considered induced mutations. So lots of different agents can cause this. There are analogs to bases, like we talked a little bit about the wobble and the adenosine nucleotide base. So that's a base analog. Things that alkylate uh, nitrous acid can lead to mutations. Acridine dyes like ethidium bromide can cause mutations. I'll explain that. I'm sure you already know that ultraviolet light can cause mutations, and we talked about thymine dimers already, and that causes xeroderma pigmentosum. We talked about ionizing radiation, right? So ionizing radiation is like X-rays and stuff like that, and those usually cause chromosomal breaks. So ionizing radiation, having too many CAT scans, this is a heavy dose of radiation. This is about 100 to 1,000 times as much radiation as you get from a dental X-ray. So the more you have these, the greater your chance of your chromosomes being broken. Normally, this can be repaired. Sometimes it can't, and if that happens in the middle of a gene that is in charge of controlling, uh, your cell cycle, so the cell cycle goes out of control, and you get all these cells growing, we call that a tumor, and that leads to the big C, or cancer. So most of these things you will probably, if you lived in California or whatever, California's really strict about environmental mutagens, and they have to label almost everything. They're considered health hazards. In a laboratory, we can use them though to induce mutations, right? And mutations help us understand the genetic content of organisms, right? So to us as geneticists, mutations are good. Without mutations, we wouldn't understand what the different genes in living systems, what their function was. All right. So this is a base analog. It's called 5-bromouracil, or FBRU. It's very similar to thymine. So during DNA replication, let's say we fed this to mice, or we fed them to fruit flies or whatever. When the fruit fly cells replicated, and then of course we would hope that this would happen in their germ cell line. When those replicated, um, this base, it's a ketone form of 5-bromouracil, and that pairs with ACE, right? So it can become ionized, right? So here, this oxygen, uh, instead of being a ketone group, a double bond, here it has a single bond with an extra electron and an extra unpaired electron. That causes the base pairing to shift. So this pairs um, with G, right? instead of A. So we have the amino group down here, which is missing on A, since this only forms two hydrogen bonds and this forms three. So in this instance, these hydrogen bonds are formed rather than these. And so now we have 5-bromouracil, which was paired with adenine, now becomes ionized and it forms base pairs with guanine. So this would be synonymous to a T, and in this case, it would be synonymous to a C. So remember, these cause transitions, right? We would convert an AT base pair, so to a GC, and then remember these are the same, these are the same. So that's a transition, not a transversion. This is just an example of it. So this is the keto form of the 5-bromouracil, and it's going to pair with adenine, right? So it acts like a T here. And then, so it's going to base pair here with an A pairing with a T like normal. And then it gets ionized to the enol form. And when it does that, it's going to pair with guanine. So we have the ionized 5-bromouracil pairing with G. And then, so in this next replication round, these strands separate. This one gets ionized. It pairs with a G. This is the original strand, so it's going to pair with the T. So after we do the next round of replication, so third round of replication, we end up with the permanent mutation, and then we still have the 5-bromouracil in here, and that could take in either form, either ionized or not unionized. But in the end, we have the mutation that's an AT now becomes a GC mutation. And remember, A to G or T to C is the transition. We can have alkylating agents that like ethylmethane sulfonate, and we call that EMS. So it can add an ethyl group to guanine, just in case you forgot. So, and hopefully you learned this in Bio 181. So this is methyl, and this is methane. Methyl group is CH3 generally, and that's bound to an R group. And then we have this is ethane, ethane, and then we have an ethyl group, which looks like this, right? So we have this bound to another R group here. All right, so here's normal guanine. Here's the ketone of this. You get an ethylated, alkylated group attached to it, which is shown here. That causes this molecule to change its shape. And so this guanine becomes ethyl guanine. And that's showing you it's on the number 6 carbon. So this is number 6. And then that causes it to pair with T's. So, G normally pairs with C, but now G is going to pair with T, right? And if we do rounds of replication, we're going to have a daughter strand that's going to pair with T or C, and then another daughter strand that's going to pair with A. So again, we're going to have G converted to an A and C mutated to a T, which again is a transition base pairing mutation. So it can this can do the same thing. It can add an ethyl group. So then this is the number 4 carbon. So it's 4-ethylthymine, which is a T. Normal thymine that pairs with G, and then so this is the normal pairing. So T would be an A, G. So we separate these, and C. So T and C, those are the same, and A's and G's, those are the same. Right, here's the G in the second round of replication. Here's the T in the second round. And so this is the mismatch. And again, AT to GC, and that's going to be another transition. All right. So collectively, uh, we say that this is an alkylating agent. Okay, nitrous acid causes deamination of the amino groups in cytosine and adenine. So this is an amino group. If you don't remember this, you can go back and look at functional groups. And so deamination, right, taking the amino group on it, leaves the ketone form of the molecule, which is uracil, right? Remember, cytosine is chemically similar to uracil. It just has this group, and then we have water. This is where the oxygen comes from. So that converts cytosine into uracil, and that's shown here. This group is cleaned off. We have an alternate base pairing. So C becomes U. U normally pairs with A, right? So in this case, we could just pretend that C pairs with A because it's going to become a U. And so in the next subsequent rounds of replication, C is going to pair with G, and then A will pair with T. Let me keep this consistent. So anyway, we have C's and T's and G's and A's. Those are the same chemical. So those cause transitions, not transversions. This is just showing you what's going on here. So we have C and G, right? This, the amino group is removed. It becomes uracil. That's what the asterisk is. It's still paired with G, which we showed. And then the subsequent round of replication, U is going to pair with A's, right? That's the normal pairing. G's pair with C's. And so you can see that the subsequent daughter strands are going to be half non-mutated and half mutated. And then, of course, we have G. G becomes an A. This is the original. And then C becomes a T. So again, these have the same, remember A's and G's are purines and C's and T's are pyrimidines. These are the same chemically related, and if we do a mutation from one purine to another or one pyrimidine to another, it's a transition. Okay. Intercalating agents are agents that slip in between or intercalate the bases of DNA. So these are things like, for example, we use ethidium bromide, which is an intercalating agent. The reason we use that is because DNA fluoresces in a wavelength that you can't detect with your human eye. So we add a compound that has an affinity to slip in the bases of this, and I'm talking about ethidium bromide. So we add ethidium bromide to our DNA. It intercalates in here. We can excite it with ultraviolet, and that it fluoresces in the orange wavelength length. So we can see the DNA. Without adding aethylene bromide, we wouldn't be able to see it because it's way below the threshold. Hopefully you remember this, but you can normally only see between 700 and 400 nanometers, and DNA is in the 240 260 range. So I'm just showing you this here. So this DNA itself, you wouldn't be able to see unless you added ethidium bromide to it, which is the intercalating agent. And then this is on shortwave ultraviolet. The box underneath it, again here, same here. This is cyber green. It's similar. It's also an intercalating agent. Uh, this is radiation. So anyway, that's how we visualize DNA. We have to use something uh, that allows us to see it in our spectrum as opposed to what DNA spectrum is which is outside of our spectrum. Um, it's ultraviolet spectrum. Okay, so let's minimize this. We talked about thymine dimers. This is just once again going. This is just a review from the previous lecture. So ultraviolet light, you're out in the sun, getting a suntan. You have two T's in your DNA next to each other. So remember, we have a sugar and then a base and a phosphate. So in DNA, this repetitive sequence is in the backbone, and so you would have a phosphate and a sugar. These are linked together, and then a base. So the bases are on the inside, and then if you looked at just the bases, it would look like this. So you would have two T's next to each other. Ultraviolet light strikes it, and then it causes these covalent bonds to form between the two T's, and that's what we call a thymine dimer. So hopefully you remember this, but covalent bonds are really strong bonds, and they're not easily broken. And so the replication for this, because these are linked together, they bulge. It causes the DNA to bulge at that point where there's these thymines next to one another. And so when the polymerase comes to it, when the polymerase comes to this bulge, it can't pass it. And so that causes the replication machinery to stop. And if it isn't dealt with, you can't replicate your DNA, and you're going to have all kinds of mutations. That's going to be bad for all the daughter cells. Ionizing radiation like X-rays can also cause DNA damage. It makes reactive oxygen species, uh, and these can damage DNA. Right? I know this firsthand. I did research on this in the VA. It deaminates bases. Uh, Ionizing radiation can remove bases from the sugar, right? So depurination and depyrimidization. Remember pyrimidines and purines. So we're just removing those, like we talked about previously. It can also break the covalent bonds between the nucleotides and generally severing the backbone of the DNA strand itself. So, like I said, you have the chromosome, the ionizing radiation like X-rays can cleave those in two, and that can cause the breakage of in the middle of an actual gene at random places. And depending on if that gene's important or not, or if it's in your germline or not, that could be really bad and lead to cancer. All right, so looks like that's the end of this lecture. Uh, so mutation one is done. I'm going to stop here, and then I'll record another um, video on tomorrow on Friday, and I'll post that. All right.